Okay, good morning to all. I'm hoping that all of you are here. Maybe one or two getting stuck in the traffic, but most of you are here. And I hope in the online participants also I can see most of them are here. So before I introduce Professor Yen Lee, let me just give you a little bit of announcement. So the announcement is there are two announcements majorly. So for the online participant, so all the wet work will start today. So far on the first 3 days, Wednesday, Thursday, Friday, Saturday, you had a in silico analysis most of the time. So you were doing computational analysis. So it was easier for you to demonstrate everything on the computer. But from today, it will not be possible. You can't do molecular biology on the computer. So you have to go to the lab to see what molecular biology is being done for particular synthetic biology applications. So in that case, for the offline participants, there are provisions for your demonstration. So you will be taken to the lab. For the online participants, what we will do is we will share a link to you. We made a pre-video. So for each day, whatever you are seeing the protocol in the protocol booklet, I'm sure all of them have been shared with the protocol booklet, including online participants. If not, please let us know. But I'm assuming that the protocol booklet has been shared with the online participants as well. So those demonstrations that will be happening on today, we will share with you the Dropbox link from where you most are not sure. Okay. So I think Brajesh, as soon as you come, I think you will share with the online participants the booklet that we are going to use, which has a detailed protocol. So today, whatever demonstration will happen in the lab, we have made a pre-video for the online participants. So we will share the Dropbox link with you after the lecture today. After the lectures, 2 lectures, we'll have a small tutorial. So in that tutorial, we'll explain to you the 3 days program that we're going to have in the lab. And for each day, we'll share you the Dropbox link to show you what are we going to demonstrate in the lab. So that has been made a video already. You can look at it. Online participants can look at it, and if they have any questions, they can ask next day. What is, whether they understood all the demonstration and all that. Okay, great. So the other announcement is that on 15th, earlier we were going to wrap up the session at 2 p.m. That's what the plan was. But I would say, fortunately for all of you and for us, Professor Ramalingam, who is going to come from Anna University, he wanted around 1.5 hours for his portion to be demonstrated. I'm sure you will love to see that, because that will be an additional knowledge. We didn't think of that. We proposed that, and I said, well, I'm sure participants are here to learn. So you might have 1.5 hours additional in the afternoon, and I hope that shouldn't be a problem. But in case you have any issue, you can let me know. But those are the demonstrations that I'm sure online participants will also be able to see, because they said we can do that on computer itself. Okay. With that brief announcement, let me welcome. So far, all of you are being welcomed by Dr. Shri Srivastava, because in the first 3 days, he is expert in the systems biology and flux balance analysis, so he was leading it. For these 3 days from today, I will be leading the session. So I'm your contact point. So I'm Shamsi Asdani, as you probably know by now, because I'm having lunch with you most of the time. So let me first thank Professor Yen Lee to be here with us and agree to give lecture. And Professor Yen Lee is a professor at the Institute of Microbiology, Chinese Academy of Sciences in China. He holds a BSc and PhD degree in fermentation engineering from Jiangnan University, also which is located in China. He did his postdoc in the institutions at Netherlands and Ireland. He was appointed as a visiting professor at Cornell University in the US, and his interest is basically in the area of industrial biotechnology with a focus on conversion of carbon dioxide into organic matters and into many useful products. And using synthetic biology approaches, his studies aimed at developing novel biocatalysts which contribute to the development of sustainable bio-based manufacturing industries. Presently, he is serving as a member of the Scientific Board of International Basic Science Program of Rubisco, and also as a member of the International Advisory Council on Global Biology. He is also a director of Chinese Academy of Science, the World Academy of Science Center for Excellence of Biotechnology. He has published more than 160 papers in very renowned journals like Science, Music Communications, PNRs, Metabolic Engineering. Not surprisingly, he has a very high agent tax of policy. So I invite Professor Yan Li to give a talk in the area of synthetic biology for carbon neutrality. So over to Professor Yan Li.
Thank you very much for this kind introduction. Let me share my screen.
Yeah, please share your screen.
So first of all, I feel honored to receive this invitation to give a lecture at this nice workshop. And I have been visiting ICGB at Trieste, I think 5 years ago. And so I know there's also a center in India and also Cape Town. So it's a great opportunity for me to have this virtual communication with colleagues working in the genetic engineering and biotechnology, which is the core site of synthetic biology, and also the participants of this workshop. And I learned from the introduction part that you are all interested in this area, and also get a practical skills from this course. I heard from the chair that in the previous 3 days, you put some in silico analysis skill, which is very important. And so I hope I could add value to you on this synthetic biology stuff. As explained in introduction, basically I am a metabolic engineer. I always give my label like this. And so I'm working in the instrument microbiology already for 16 years. So carbon neutrality is one of the trending topics recently across the globe. I want to illustrate how synthetic biology can do for contributing to the carbon neutrality. Apparently, this is not our major job, because currently cutting the CO2 emission from the transportation is the major resources for achieving the CO2 peak. But you're all interested in synthetic biology. But when I always talking to synthetic biology to the newcomer, I always using these slides. So I found there's a limited understanding from the public conception on synthetic biology. So if you talk to the public to any people with different various backgrounds, about 1/3 of the people will think synthetic biology is unnatural, so man-made, something that isn't real, artificial. Those are the most received response from that. But there will also be something talking about cloning, for instance, genetic manipulation, and something synthetic oils, and some medical diseases, all kinds of different answers you will find. And certainly there's also quite high number, 1/4 of the people don't know what is synthetic biology. So synthetic biology is really new, although for the genetic engineer, it's not new, because if you go back to the origin of synthetic biology, could go back to the 70s of last century, where the first genetic engineered insulin produced by yeast could be considered as early example of the synthetic biology practical application. Now, it's what I define synthetic biology is a more advanced technology for engineering the organisms. So we certainly need to give them a definition, and people always say synthetic biology is a new frontier of biology. But what is synthetic biology for? Basically, there's two main strands. So one mainstream of the synthetic biology research is to design and engineer novel biological parts, devices, and systems. This can be a promoter, and could be and also could be an enzyme, and could be an enzyme cascade, or could be artificial cell. We know the synthesis of artificial cell from scratch has not been realized, but we could utilize ATCG to synthesize a chromosome and to replace the chromosome of existing microorganism and make it alive. So we are hoping that in the future we could starting from ATCG from the scratch and building a live cell, and there are many scientists are working on this direction. So relatively easily is the next stream, which is redesign existing biological system for useful purposes. And that is the metabolic engineering for. So you could engineer the cell from different degree. You can modify one gene, you could modify multiple genes and accept a set of 20 genes or even 100 genes of a microorganism, and redirect the metabolic flux and to the useful purpose, for instance, production of fine chemicals or even bulk chemicals. So these two streams actually summarize the basic direction of the synthetic biology. The first one is more difficult, and the second one is realizing. And even on the industrial scale, some engineered organism has been put into practice for production of certain chemicals in a cheaper way. So how do we do that? Is basically studying its engineering concept is very important. So in the States, it's also called engineering biology, because some people, because of the religious, perhaps the reason, they don't like the word synthetic biology. So in the US, they do call it engineering biology, but usually we could make it equivalent. So it actually combines many different disciplines. For instance, the bioinformatics you already touched a bit in previous days. And chemistry, certainly we need to understand the basics of the biochemical reactions. And the mathematics, often the biology students does not really have affinity to the mathematics, but modeling do need mathematics and computing skills, as you have learned a bit. So we need to incorporate the engineering principle, for instance, modularity modules, and abstraction. We want to abstract the basic life principle from a highly dynamic lives, and we also want to create some orthogonality into the classical biotechnology. And this orthogonality means in the living organism, it's a highly complex system. We want to put into, for instance, a genetic engineered pathway which does not interfere with its existing pathways. This is a kind of orthogonality. So those modularity, abstraction, and orthogonality is not the concept that we learn from the conventional biology, and those are the concepts from the engineering part of view. But they need to be converged with multiple disciplines so that we can engineer the cell according to our demands. We all feel that synthetic biology is very hot, is a trendy topic. From the commercial point of view, actually last year, globally, we have seen more than $100 million investments. So why this is the point we are talking about synthetic biology now, not 10 years ago, not 10 years later. And this is actually because synthetic biology has accumulated its technology breakthrough to the abundant number, so that we can do a lot from the synthetic biology. It's actually starting from 1953. We have the DNA structure resolved. That's the basis, fundamental basis for we can perform genetic engineering. And we all know that from the 70s last century, the first example of genetic engineering emerged. And then we have the Human Genome Project completed in 2000. By that time, sequencing a human genome, 3 billion bases, we need loads of money. But now, the amount of money for sequencing a human genome is now about $100. and it could be even lower. And for sequencing a microbial genome, now it's becoming a routine practice in many laboratories, including our lab. So for sequencing a microbial genome with, for instance, 2 million or 4 million base pairs, it just costs maybe, I think, maybe a sequencing human genome is $1,000, and sequencing a microbial genome is just $100 or even less if you sequence more. So sequencing price is very low, and gene synthesis price also decreased significantly. 10 years ago, when we want to synthesize a gene from scratch from ADCG, it's basically we need to pay about $10 a base pair. But now we only need to pay, I think, let me think, it's 10 US dollar cents, 10 cents for a base pair. It's predicted that the cost could reduce to 1 cent, I mean US dollar cents, for 1 base pair. So at this stage, our laboratory and many students, if they want to save time, they do not do PCR anymore. They just send a sequence to the company and get a plasmid back with the sequence of your desired gene already on the plasmid, and do the transformation, do the integration, etc. So we would foresee that synthetic biology is becoming a very routine technique in the laboratory to use. And PCR may only be used to demonstrate or to confirm the gene knockout or gene integration, etc. So the significantly decreased genome sequencing and gene synthesis cost actually opens a novel way to understand the principle of the life organism and create a paradigm for biological research. It actually completely changed the landscape for doing biological research. There are many areas to be described in synthetic biology. I just listed a few, and this is actually expanding. For instance, we have people working on synthetic genome, we all understand that, but also people working on the minimum genome. And people try to reduce the genome size to see what is the core genome for conferring the basic life requirements for a given organism. And for E. coli, we already know it's 4 megabases genome, million bases genome. It can be reduced to 40%, but they're still alive. Showing multiple genes, many genes in the genome is redundant at certain circumstances. I already explained the orthogonality, so there are many researchers working on the orthogonal biosystems. So putting, for instance, not NADH, NADPH, it could be XADH. And people also studying the xenobiology, which means you could incorporate not natural base, ATCG, but it could incorporate a man-made base into the chromosome, but the organism is still alive. And this could add the cell additional capacity that they do not have naturally. And many people working on the regulatory circuits, and if you are also participating in the IGM competition, and there are many projects working on the circuits, genetic circuits, and the genetic circuits could mainly used for a signal transduction or for controlling the DNA device and parts to work on a certain regular rules. And there's many people working on metabolic engineering because this is the closest area that can engineer the cell towards useful purpose. And there are many products can be produced. It could be bulk fermentation products like amino acids, organic acids, biomaterials. It could also be many fine chemicals, for instance, carotenoids, etc., from those products originating from plants. And there's a niche area called Proton Cells. These Proton Cells, researchers are interested to understand what's the original cell look like. So we have the E. coli now, which we know they have survived for many years. But there are also cells extinguished. You could not no longer find it now. but they present a million years before. And you could not easily identify in the fossils, because for the fossils, easily you can identify large animals, large plants, but for microorganism, which is not visible using your eyes, but also difficult to find in the fossils. Can we predict from the genome sequence that we have now, so what could be the proton cells, and get it alive in the lab by different technology of synthetic biology? And there's also a niche science area called the bio-nanoscience, for instance, to combine the life organism, life material with nanoparticles. And there's already a lot of research on using nanomaterials combined with the microorganism to increase the photosynthetic efficiency. For instance, using many nanomaterials to capture light with a high efficiency than the plant itself, and transfer the light, transfer the photons into microorganisms, converting them into reducing equivalents, NADH and ATP, to support the growth of the organism. And this is also a very interesting area for that. Those are the main areas that synthetic biology mainly cover, but many people also included the genome editing, the gene editing, into synthetic biology. That's an enabling technology. I don't have any objection for that. But in terms of the area with different applications, and those are the main areas for that. With synthetic biology, with the creating ability of synthetic biology, it's actually breaking the life boundary, starting from 12 years ago, the first synthesized genome from Craig Venter, putting synthesized genome and putting to a microorganism and making that organism still alive. And I said xenobiology, we could organize an unnatural base, incorporate that into the RNA or into DNA for that. We have 20 amino acids we know that is composed of the proteins, but we could have many unnatural, non-natural amino acids being produced in a lab condition, and incorporate that into the peptide or into proteins, and E. coli is still happily living. And we could also creating novel function for ribosomes, is not only for translation, but it could also having some other functions. So there are many, many more new. We are actually breaking the life and non-life organism. This interface is no longer that clear because of the synthetic biology. One of the big consortium project is the synthetic yeast chromosome. We know yeast has 16 linear chromosomes. And so there's a consortium people from US, China, UK, France, Australia, and Singapore. And they have just building from scratch and synthesized different size of the yeast genome and replacing the linear genome, linear chromosome in the original yeast. And this is actually a quite tough job, but they made that. And during this project, they could also fix some unnecessary DNA fragments in the yeast chromosome. So this is a very ambitious project being achieved. And they found the yeast with the artificial chromosome, the de novo synthesis chromosome, did not really differ in growth and in the stress resistance to compare with the original yeast. And there's another interesting project on yeast chromosome is Professor Zhongjing Qing, my colleagues in Shanghai Institute of Biological Sciences of the Chinese Academy of Sciences. He did an interesting project. We know yeast chromosome contains 16 linear chromosomes, but he actually connects this 16 linear chromosome into a circular chromosome. So this is a linear chromosome organization in the configuration in the yeast cells. And when it's head to tail connected into a circular chromosome, you get the chromosome like this one, but it's a supercoiled. This is a structure you can call single chromosome cells. And he demonstrated the single chromosome cells that connected by these 16 linear chromosomes is still alive in the cell. So this is actually very interesting. But the cells, yeast cell with a single circular chromosome shows a little bit defect in the different stress, and also they have not found what's the usage of this cell within a chromosome yet, but this demonstrated nature created stuff could be changed in the lab. They just demonstrated this rationale. So with many breakthroughs of the fundamental research of synthetic biology, the potential of unlimited potential biology has been created. For instance, just name a few examples. The breaking cornerstone of the artemisinin, and which is used for creating the curating of the malaria, can be produced by yeast. Yeah, and Taxol, which is an anti-cancer drug, can also be produced. And Alcan can be produced, but in a very low yield, so it's not in a commercial scale yet. And we could also take up CO2 and use inject electrons from the electrode that is actually directed by electric energy to fix CO2. So this will be more efficient compared with using the light as the sole energy source. And we could also change the conventional treatment for hypouricemia and many diseases. So what we could think about synthetic biology is a potential application in healthcare and drugs, environmental, is many things. To my understanding, the emergence of the synthetic biology could reshape the Earth on the way we obtain the resources. So think about in ancient times, we get plants and animals for our survival, for our life, and we brew different fermented foods and alcohol from starch, from soybean, from vegetable, and from fermentation of the meat. That's for human life. And when the penicillin fermentation about 80 years ago went into practice, so we started to produce the fermentation products, including antibiotics, vitamins, amino acids, organic acids from sugars, to satisfy our needs, and those are industrial products. But nowadays, because of synthetic biology, we no longer we could use corn starch to produce those kind of industrial products in a more efficient way. But we may also no longer need the existence of the plants. We can directly utilize CO2 with energy from light or from electricity, and to create stuffs that originally we need to extract from plants. So by this way, we could save the land for planting. We could even in the future, we all heard a lot, to produce meat in an industrial way, so we don't need to feed cattle, and then we could also save the CO2 emitted by those cattle, etc. So synthetic biology actually opens a way that you can rethink of the way how to get resources from the nature, and how to preserve the earth, and how to change the climate change situation. And even in the future, how can we produce food for us if we want to travel to Mars and to the stars that is outside the Earth, or even the solar system? And that's why what we are interested in, as we said, traditional, conventional, we are based on the fossil fuels. So this is basically fossil-based fuel and fossil-based chemicals. And this is not carbon neutral, because the fossil resources are formed by millions of years. And basically they are from the biomass, originated from trees on the surface of the Earth. By millions of the geo-activity, and they are condensed, become highly dense energy resources to support industrial running. And when we take it out, so certainly we get fuels, we get a few our cars, we get our chemical industry, we're getting our plastics, our fibers, and every stuff are based on the fossil fuel resources. And certainly we get some CO2 released from using the fuels, and when the life cycle of the fuels, material, chemicals finish, they also become CO2 released into CO2. That's why we see a very rapid increase of the CO2 during the last 200 years after the Industrial Revolution. So to achieve a carbon neutral society, we have to gradually reduce our dependency on using fossil fuels. And they have to be underneath the earth, underneath the soil, do not take it out. But using the carbon dioxide already in the atmosphere, and using it as a raw material and to produce those fuels, material, and chemicals, so that this carbon neutral society can be achieved. So in the future, we are only using the we call the present carbon, which is already in the atmosphere, but not using the older carbon, which is underneath the earth. So this is the idea, but certainly because of the emission of CO2, large amount of emission is not formed in one dice. We are not expecting we could achieve the carbon neutral in one dice. So all the different countries are putting this target for next 30 to 40 years, even longer. And so how synthetic biology could do that? So we could design engineer cell factories for what we call the bio-based manufacturing, so that we can, using the sugar as a raw material, or even using CO2 as raw material to produce hydrocarbon and petrochemicals, and also even natural products that originally we have to extract from the plants. There are many, about 2 million different natural products can be produced, so giving the huge market potential for synthetic biology for that. And for achieving those goals, what we need to do is to engineer the cellular metabolism. It's very complicated, but it's engineerable. And for the biological conversion of CO2 that we are interested in, it's not a new topic. For chemistry, we already can, globally, we could produce about 20 million tons for urea from CO2 and ammonia, but this is energy-intensive production process. And nature also does the job because we have many trees and algae in the nature, and also the crops. They actually utilize the sunlight and take up the very low concentration of the CO2 in the atmosphere, and they grow using a few months, and they supply the food for us humans. So usually, for instance, the crops takes 3 to 4 months and to get it ready. In general, they have a very low efficiency, but because the sunlight is free, the land is free, water is free, so the crop has relatively low prices. But a low price does not mean the value is low. When you have the war, those things will be very expensive. So the price does not equal to the value. So mimicking that process, when we want to utilize CO2 in the atmosphere, we need to inject energy. Basically, if we could increase the density that we inject into that cell, for instance, not the low density light, but the high density electric energy, that by principle, we should be able to utilize CO2 in a more efficient way. So we are hoping to develop an efficient biocatalyst that could be 10-fold higher than the plants, or even 100-fold than the plants. And process completed not months, but within days or weeks. So we could commercialize this process not using the nature's power, but using industrial power. So we could convert or transform originally the natural months-long process into an industrial level to a days-long process. Then we could change completely the production mode and also the development mode. One example that we published on the Biotechnology Advances is to develop, to engineer the algae. Algae, including the cyanobacteria, algae usually is called eukaryotic cells, but cyanobacteria is prokaryotic cells. There are many researchers in the world doing that job. So by redirecting the metabolism of algae, including cyanobacteria, we could utilize CO2 as the sole carbon source to produce a variety of different chemicals, including, for instance, ethanol, ethylene, acetone, isopropanol, those are C3, and lactic acid, glycerol, and butanol, etc. Or even longer one, farnesene, alanine, fatty acid, and even sugar, sucrose, and also glucose. But the yield is very low, just because about a gram per liter to 10 gram per liter, so it's not reaching a commercial scale. And also the time is quite long, about 1 week to 2 weeks or even months. Because the cyanobacteria is still considered, it's not a plant, but the energy efficiency for utilizing sunlight is still low. How do we do that? Is we engineer the light conversion mechanism and also the carbon conversion mechanism of cyanobacteria. So basically, we have the solar photons going to the light reaction system, and the light reaction system converting the photons into the NADPH and NAATP, and feeding to the carbon cycle using the Rubisco, which is a key enzyme, maybe the most abundant enzyme on Earth. Every plant has an enzyme and fixes CO2 into 3-phosphate glycerides, entering into glycolysis, and then channeling to the different metabolism. And if you want to produce a target chemicals, you want to delete the competing pathway, you want to delete the storage pathway, and then with this you could direct the carbon flux into the targeted chemicals. The targeted chemicals can be any chemicals of your desirable feature. And you also need to overexpress, for instance, the key enzyme to magnify the flux from that. That's the basic principle of metabolic engineer. Compare with E. coli or yeast, which is only engineered cut to paste, to cut the competing pathway and enhancing the core pathway. For the cyanobacteria, those photosynthetic organisms, you also need to deal with the photoreaction, which is a very complicated system. So leading the engineering practice is getting more complicated. I talked a bit about Rubisco, and so some just examples that we did in our lab. Rubisco, with the full name, is called 1,5-ribulose biphosphate carboxylase oxygenase. So this is a very ancient enzyme. In terms of the protein content, this enzyme is the most abundant enzyme on Earth. You can find it in every plant, in every algae living photosynthetic, using photosynthesis to grow. Rubisco is a very complicated enzyme, but with a very low efficiency, with a turnover with only 1 per second. So you mean every second is only 1 reaction can be catalyzed. What is this concept? Consider the industrial enzyme. It could be 1,000. So 1 second can catalyze 1,000 turnover of the reaction. So Rubisco is a very lazy enzyme with very low efficiency. And there are several forms of Rubisco. The Rubisco form 1 is the most ubiquitous Rubisco. All the plants has in the form 1 Rubisco. It's very complicated. It has 8 large subunits and 8 small subunits. So making it very difficult to engineer or even very difficult to express. So that's one of the reasons why engineered plant and photosynthetic bacterial Rubisco is very difficult. And there's also form 2 Rubisco, which is much simpler, only large subunits and only a dimer. A dimer could be a multiple dimer of that, but compared with the form 1, it's much simpler. And there's also form 3 Rubisco. It's also only 2 dimers of the large subunits, but with a different configuration compared with the form 2. And there's also form 4 Rubisco, which is called a Rubisco-like protein. They are found in archaea as well, but to date, no activity of the form 4 was detected as Rubisco. So basically, it is only form 3 available. We are not working on the form 1 because it's too complicated, but we are actually interested in the form 2. Many people working on the form 1, which has making some breakthrough. For instance, you could put into the carbon cycle, which utilizing Rubisco as a key enzyme, and feeding the formate as a co-substrate, and to support the growth of E. coli cells with a very long doubling time. Because E. coli, we know the fastest doubling time is 20 minutes, and this one using CO2 and formate as the sole carbon source, it takes 20 hours for doubling. And you could also introduce this carbon cycle into yeast cells, making the yeast cell to grow on CO2 and the methanol. The methanol certainly provides some energy, and with very low specific growth rates and doubling time of 40 hours, because we know for yeast usually they could double in 40 minutes or slightly longer. But those are breakthrough of using the natural Rubisco from form 1 from the plant for achieving autotroph growth. What we found was interesting called a tubal worm, which live in deep sea. Deep sea, we know deep sea there are some volcanoes. So in deep sea there are some places which is rich in CO2. So we discovered a very interesting Rubisco in this deep sea worm, and they grow very rapidly. And there's no organic source available, only inorganic source, for instance sulfur nearby. But this worm called a Rifita, they grow very rapidly, and because they contain in their gut a Rubisco enzyme. So we isolated those Rubisco enzyme and found it has a very high activity compared with the cyanobacteria called 7002. So we put them into E. coli. We call it RPE Rubisco, form 2 Rubisco. They could express to a very high amount in E. coli, and could also show very high concentration of fixing CO2 compared with the cyanobacteria for that. So if we name the form 1, which is here in the Rubisco soluble expression and also carboxylation rate, you see the form 1 from green bacteria, form 2 from red bacteria, they're actually very low. And our Rubisco isolated from the tubal worm in deep sea, they actually outcompete of all existing Rubiscos, so making this Rubisco a very competitive enzyme for subsequent use. We actually, based on this newly discovered Rubisco with very high activity, we constructed a pathway for catching CO2 and producing, using xylose as a co-substrate to produce lactic acid. And we demonstrate actually with this RPE Rubisco from deep sea, they could produce more C13 labeled lactate compared with the cyanobacterial Rubisco, about 4-fold higher for the higher carbon capture efficiency. So demonstrate in vivo the usefulness of this new Rubisco. We could also use these Rubiscos another feature to produce glycolate. I mentioned Rubisco has two functions. One is carboxylation, but the other, the oxygen, could also compete with CO2 for the active site of Rubisco. So they can add CO2 together to go to, for instance, the carbon cycle, but the oxygen could also react with the RPE to produce 2-phosphate glycolates, leading to the glycolate production. So we are not only utilizing the carboxylase activity, we could also utilize the oxygenase activity of the Rubisco. So using this form 2 Rubisco, we could, using CO2 as raw material and oxygen as a material which is abundant in the nature, and we could produce glycolate from CO2. This is the data obtained from the lab. But the amount is not very high. We could produce about a few grams per liter of the glycolate. Glycolate is also important, useful chemicals used in the chemicals industry. And the next example we are talking is, we talk about photosynthesis. Photosynthesis is a very complicated system. So you have photosystem 2 and photosystem 1, and the converting photons into NADPH, and using the membrane potential to generate ATP, and etc. So the hydrogen are actually from the water releasing the oxygen. This is the basic principle of the photosynthesis. So basically, there's electron flow on the membrane, and that is where the photosynthetic enzyme resided. So that's electron flow is starting from the photosynthesis. So we ask the question, can we consider using the photosynthetic electrons into electricity? So basically, if we generate sunlight, we could using an anode to extract the electron, and if we have a cathode to be incorporated, we actually could form electricity. This is what we call the biophotovoltaics. We all know the photovoltaics is actually using semiconductor materials to convert sunlight into electricity, but the photovoltaic material is harmful to the environment. If we could using a live organism to convert sunlight into electricity, that will be cleaner and more sustainable. And so we actually investigated the literature. There's already many people doing that. But due to the difficulty of transforming the electrons outside the membrane and cell wall, which is insulative, the electron is very difficult to be pumping out from the cell wall. So you could see that for the photovoltaics, large scale, they could achieve 10 volt per square meter. But for the biovoltaics, it's actually 1 magnitude or even a few magnitude lower compared with the currently used photovoltaics, meaning although many researchers doing this work, the efficiency is very low. So the difficulty is if we're using single photosynthetic organism, we are difficult to get electron out. So we come up with a microbial consorting idea. Say we convert the sunlight, the energy, and using the biomass to convert into a mediate we call the energy carrier. And so we're using CO2 and we fix CO2 and producing lactate. Then we use another electron generating bacteria to consume the lactate as an energy carrier to produce electrons. And we know there are many electricity generating bacteria can do that job. So the question is now converted to how can we get the photosynthetic organism and electron generating organism work together efficiently using sunlight as the sole natural energy source. So then we hoping by doing so we could put in a called a Cyanobacteria, which is one of the cyanobacteria, and the other is Schwannella, which is a gram-negative bacteria, which is electron generating. We could build up a consortium like this, taking up sunlight and converting the electron into the lactate, fixing CO2, and let the lactate penetrating into the Schwannella organism and get electricity generated and taken up by the electrode, forming the electrical current. So in this way, you could have a direct way for creating a photoelectricity conversion. And those are the details for the study. We are not going to talk about that, but there's many control for only Schwannella, only cyanobacteria, but basically a mixture of the both strands achieving the highest electricity generation. And we could also put in a system and let it run for 40 days, so generating a power of 130 millivolts per meter square. This voltage can actually lighting up a very small bulb for that. So this is actually the longest biophotovoltaics reported to date. For previous one, they only can last only a couple of minutes or only longest a few hours. So meaning that if we could further increase the photovoltage by 10 times more and can still lasting for 40 days, these biovoltaics can be put into practice, for instance. And we could also test the stability, and we use different mediums and different Schwannella for 1 to 20 volts. They can stably run for that. And we could also see that the cyanobacteria growth in this setup is also promoted. And so the cell wall insularity is the original problem for preventing the electron to be pumping up. So by designing the consortium, we actually circumvented this problem, increasing to a tenfold higher voltage we produced. But ultimately, we're also still working on how to get this electron pumping out. And so our lab is still doing another approach for not using the consortium approach, but still using the single organism because it could be simpler. And so by doing that, it's a cartoon like this. These two microorganisms work happily to convert microorganisms to convert sunlight into an electricity. Next story, I'm going to talk about the carbon fixation. And this is a complicated this carbon fixation cycle, which we found in nature. And this first one is carbon cycle, which you assess in most plants and cyanobacteria. And the other one you could also find in different bacteria, and some are not commonly found. And this linear one is very interesting. You can find mostly in the Clostridium And there's a company called LanzaTech in the US using CO from steel mill off gas to produce ethanol, basically using this called WL pathway for that. One common feature you can find from natural carbon fixing cycle is very long. They usually have more than 10 reaction steps. Some are longer, as long as 16 steps. So in 2016, there's a research group in Max Planck Society Institutes, and they actually developed the first called a Setch cycle. This Setch cycle is not present in nature, but is a completely new cycle. It's shorter than one of the cycle, but it still contains about longer than 10 reactions. And this was on Science and reported as the first synthetic cycle, and I wrote a perspective on those cycles. But we asked the question, can we construct an even shorter, a minimized carbon cycle? So we come up with a research design and minimize the carbon fixed cycle. What could be the minimized number? So the minimized number could be 3 steps. Starting from C2 carbon, and adding one CO2, getting a C3, adding another CO2, getting a C4, then getting maybe something 2 carbon split out, so getting this cycle. So theoretically, the minimum carbon cycle should be containing 3 actions, but practically no such reaction is available. If we starting from acetyl-CoA, add a CO2, we could get a pyruvate back, and we add another CO2, we could get an oxalic acetate. But we cannot turn oxalic acetate into acetyl-CoA using one single reaction. So we add an additional reaction, which is acetate. So this enzyme could convert oxalic acetate into acetate and releasing an oxalate. And then we have another enzyme could convert acetate into acetyl-CoA. So then we develop a design which is a 4 reaction mixing 2 CO2 and creating 1 oxalate. So that's the overall reaction. And we calculated the Gibbs free energy, and we found it's actually generate negative, meaning this cycle has the driving force to get it wrong. And the next one is how can get it wrong. So we have this overall reaction to have carbon dioxide and fixing with reductive ferrotoxin and ATP injected. So we also selected 4 enzymes with pyruvate synthetase to convert acetyl-CoA into pyruvate, and pyruvate carboxylase into pyruvate to oxalic acetate, and oxalic acetate acetyl-hydrolase, converting the 3rd reaction, and also the acetyl-CoA ligase into that one. That's from the biochemical database. And this is a long way, to cut a long story back, we needed these 4 enzymes. But next question, where can we get this active 4 enzymes and get them 4 work together? So a lot of screening process being performed. And the most difficult one is this one, the pyruvate synthetase, also called the pyruvate ferrotoxin oxide reductase. This reaction is actually usually catalyzed the 4th reaction, which is catalyzed pyruvate decarboxylase. So to CO2 and acetyl-CoA is more favorable for that. So if you want to condense CO2 and acetyl-CoA into pyruvate, the Gibbs free energy is positive, meaning this reaction is very difficult to occur. So you do need an electron donor, ferrotoxin, and very reduced form to drive this reaction. And this reaction, ferrotoxin, is actually has to be run at -500 millivolts. So this is a very reductive environment, meaning that you have to be very anaerobic with a very active ferroredoxin to drive for that. Fortunately, after years trying, we have identified several ferrotoxin with quite high activity, and we could run this reaction at 50 degrees to drive acetyl-CoA and CO2 into pyruvate. Because pyruvate, this reaction is revertible, so we have to using lactate dehydrogenase to drive the lactate into pyruvate into lactate. This is a very high driving force, so that we can measure the activity, how much pyruvate is produced by using the C13 labeled technology. And so we get this done, but this is actually the most bottleneck part. Then we got a pyruvate carboxylase screens from a certain strand. We get a 50 degree. Although this one is more active, but it's 80 degree, which is not compatible with our 50 degree temperature. And we'll get another one, oxaloacetate acetalhydrolase selected, which is very high activity at 50 degree. And the last one, the acetyl-CoA ligase, we also screen different resources, and we get one from 50 degree quite active. And then we get all the enzyme required. Mostly are active at 50 degree, but with that one is only available at 28 degree, which perhaps they will lose activity at 50 degrees. So we need to replenish this enzyme, like original doing PCR reaction, you need to add enzyme regularly. So those are one PFOR from Clostridium and OH from Aspergillus, and the other is from some archaea. So this is actually mined biodiversity from the genome database, and you get all the possible enzyme for assemble this Pope cycle. And for assembling this Pope cycle, we did it once, but not easy. So we cut this Pope cycle into two cascade reactions, and we test these two cascade enzymatic reactions. If it's working, then we assemble this cascade reaction together, and we put it into the work. And finally, we get actually this using C13 labeled. We put these two reactions together, and we get actually the C13 oxalate produced in a time course when we only injecting C13 labeled CO2. So meaning that the Pope cycle is functional. And so the constructed Pope cycle is to date the minimized synthetic carbon fixing cycle compared with more than 10 reactions. It only contains 4 reaction steps. Certainly the efficiency is not very high because the PFOR is really a bottleneck, but providing a possibility to expand from this scratch. And the last slide I want to show is a work that I was involved, but I was not a corresponding author. This is actually last September in Science. Is my colleagues and we developed a completely novel pathway for converting CO2 into starch. And this actually called a chemoenzymatic starch synthesis from CO2. The first reaction is CO2 using hydrogen and a chemical catalyst to produce methanol, and then using 11 enzymes to convert the methanol into starch. So this is the first non-photosynthetic synthesis of the starch, and making it possible for the future to directly synthesize starch from CO2 rather than for planting crops using the fertile land for that. At the moment, the cost is still very high because the enzyme cost and also the reaction cost, etc. But given the conceptual breakthrough, people can think when the enzyme is cheap enough, when the efficiency is high enough, you can expect in the future that one day the production of starch from industrial scale can be achieved rather than from agricultural modes. So we could expect in the future when you have a sufficient low energy from electricity or from the hydrogen, when they are cheap enough, we can use CO2 as raw material to produce a variety of different chemicals that is now produced from the petrochemicals, so making a significant contribution to the carbon neutrality. So I think I'm running out of time. Concluding remarks is we can feel that synthetic biology is blooming, but for achieving the standardization and dynamic simulation and also the interactions, still a long way to go. And one single group is not possible to do everything. For the starch project, we have more than 10 groups and more than 100 people to be involved and taking 6 years. And for my postdoc, this is only one PhD student work, but it also take about 5 years to complete this very tough job. And there's a lot of needed for the industrial academic collaborations, and also the global collaboration is very important. So finally, I want to acknowledge my students for the Rubisco. My PhD students, Junmi Zhang, just graduated, did a fantastic job. And Oxide is my PhD student, Yang Fan. And Biofuel Photovoltaics, Huawei Zhu, who is a PhD student, postdoc, is still developing a new generation Biofuel Photovoltaics in the lab. The Pope cycle was done by Xiao Lu and spent 5 years' time. I also acknowledge the funding from Natural Science Foundation of China, Chinese Academy of Sciences, and Ministry of Science and Technology. And finally, I'd like to thank you for your attention. thank you for implementation. I'd like to take questions if there is any. Yeah. Very good question. Actually, at this stage, none of the POP pathway or the SAP pathway is commercially viable. For the POP pathway, it's still what we call is a 1.0 version. Because I said the PFOR, the pyruvate synthase, is really a troublemaking enzyme. My students spent about 3 years to tackle this problem. So when she is close to her finishing time, she has no more time to further optimize that. So that which is a pity. So we at this stage we demonstrate the feasibility. Actually, the POP cycle is wrong, but it cannot run a few cycles. So for future work, we need to have a PFOR with higher activity, which could drive the cycle to go further. But even if go further, the oxalate is not a very expensive pathway, very expensive products. So economically, it's not viable. So the significance of the POP pathway is we demonstrate scientifically that the carbon fixation cycle can be short as 4 reaction. For the ASAP pathway, it's already the version 3. For the version 1, it's actually demonstrated. We see the starch blue actually in the lab about 3 years ago, and then they optimize the enzyme in some key reactions, get the ASAP 2.0 version, and then they connected with the CO2 to methanol pathway, get a 3.0 version. With this version, they now achieve efficiency of about 8-fold higher than the photosynthesis, but still not enough to achieve production on a commercial scale. So if we say the price of starch is $1,000, then about the starch synthesized in the lab may be 30-fold higher, more expensive that. So there's still a long way to go. Again, it's demonstrating the scientific possibility. Yeah, I did not go into detail. We know that presently the highest activity for the Rubisco is from the cyanobacteria. It has a turnover number of about 14, compared with about 2 or 3 compared with the Rubisco from plants. So the activity is the turnover number from cyanobacteria is high. What we have the one from tubal worm in deep sea. It shows a turnover number of 28. Which is double the activity of the one from cyanobacteria. But most importantly, it is very easy to be highly expressed in E. coli. So that's making the raw activity of the overall Rubisco is very high. So the specific activity is only 2-fold higher. But since it is readily expressible in E. coli because it only has a dimer, compared with the 8 large subunits, 8 small subunits compared with the form 1. So the core is that. So we think leading to the overall activity 10-fold higher. But the application of Rubisco in the cell is still troublesome because overall the activity of Rubisco compared with the other highly efficient carboxylase, for instance the PEP carboxylase, the phosphate carboxylase, which is about 100-fold higher than the carboxylase. So making the artificial carbon cycle is still difficult. The reason is that in the nature, for plants carbon cycle, they don't need to be very efficient because the CO2 concentration in the atmosphere is low anyway, so they just need a month's time to utilize a very low concentration. So this is perhaps kind of evolution. The nature evolved a low activity of Rubisco to adapt to the low concentration of the CO2 in the atmosphere.
Thanks, Professor Lee, for the nice presentation. I have one question about the stability, like when we make some new pathway and sometimes we need to, after some days or years, we go the large scale, it will get back in the natural way in the previous one. So we have to remove some key points by the CRISPR-Cas, so it will maintain that pathway. How? What do you comment on this?
It was not very clear, but the question is when you want to engineer the pathway, so what's the rationale to remove pathway and enhance pathway? Is that your question? It's a very good question. Usually, the cell does not to have new proteins because the new protein introduction actually interfere with its own existing physiology. So when you try to let E. coli to express a protein to a high amount, it's not happy. But our mission is to make the microorganism, the chassis that we are engineering, they are a little bit silly, so they do not feel they are overexpressing a certain protein that is not belonging to them. So which means, for instance, some Clostridium, they originally have the restriction system, meaning that any foreign DNA, when entering into the cell, the restriction system will cut them into pieces. That's the reason why you cannot transform DNA into many wild-type organisms. That's one common feature. Well, once you understand the problem is because of the restriction system, you can actually mimic the restriction system. You can, for instance, do methylation. You can methylate your foreign DNA. The methylated DNA, when transformed into the host, it will not be cut by the existing restriction modification system. So there are many different organisms, many different mechanisms for helping the host for preventing foreign DNA entering. For E. coli, since it's already been well studied and it's all barrier for hampering the foreign DNA entering have been removed, so it's relatively easier. And nowadays, when you want to achieve a stable expression of heterogeneous proteins, you can easily integrate into chromosome even without any plasmid. We have achieved so, for instance, several years ago, we engineered a complete chromosome-engineered E. coli for producing butanol. For that strain, we deleted more than 20 genes of E. coli own chromosome, and we also inserted more than 15 different genes with different copy numbers. And so the whole yeast strain does not have any plasmid, so all the foreign genes is integrated into the chromosomes, and they are just stable. So during the heritage, they consider this is part of their own machinery, and they are adapted to it. So we also see, for instance, the alanine and also succinic acid engineered E. coli already being able to produce those chemicals in industrial scale, and those are all engineered, chromosomally engineered, and we have never heard the degeneration of those strands. And so it's not a problem already for well-studied organisms. So that's why the synthetic biology, they are calling for the standard chassis. For E. coli, is the Saccharomyces cerevisiae, Corynebacterium, Protomincan, and Bacillus subtilis. And they are very well studied. They are, as I said, they already been studied, so they can be controlled by the human being.
Hello, sir. Am I audible? Hello.
Yes.
Okay. So it was a great talk. I just wanted to ask one question. If we want to produce any antibiotic in E. coli, and after production, will it not lead to the death of the host organism itself? Like, for example, I want to produce any beta-lactam antibiotic in E. coli. So, will it not affect the growth of the producing organism itself? And how do we overcome this?
The question, if you want to produce antibody in E. coli, then I was not clear. Will that antibody be tolerated by E. coli? Oh, antibiotics, okay, yeah. Yeah. Yeah, it's unlikely. So that's why people often using actinomyces, which is antibiotics native producer, to produce that, because it's like for the antibiotics producer, which is often actinomyces, they often can produce multiple antibiotics. Antibiotics are often organized in a gene cluster, and gene cluster usually contains one immunity protein. That's why actinomyces will not be killed by the antibiotics it produced. So if you want to let E. coli to produce, it's also fine, but be sure you understand what's the immunity protein of that antibiotics, and putting that together with the other gene cluster into E. coli, then you may be able to produce the antibiotic with not killing the E. coli for that. Yeah. But E. coli is not antibiotics good host, because I have never heard using that apart from the research for that.
Hello. Am I audible? Okay. Thank you, Doctor. Indeed, it was a nice talk listening to this thing. So actually, I'm more of a systems biologist, where I do more of a computational work. I do genome scale modeling, I do flux balance analysis. So I'm quite new to this field, and I just have a basic question. So I would ask you what standard textbook or some classic paper which you recommend to me. And I also work on cyanobacteria, so I was interested in some part, but I'm just like quite new to this thing. So could you suggest something where I can learn these techniques which I can implement in my research work?
Is the question about what's the classical paper on system biology?
Papers on, yeah, because I'm quite very new to this field. I only work more on the systems biology part, and my work is more. Yeah, from scratch. So you could just.
That's a very interesting question. There may be so, but I have not read. I only know one of my colleagues has written a Chinese version, so I could not recommend to you. Well, there may be, because the one written in Chinese is authorized in to be used in the higher institution in China. But I did not coming to see what would be the English version. Yeah. Yeah.
Thank you. Thank you.
Good morning, Professor Lee. Thank you for the very wonderful presentation. My question is that when you are expressing Rubisco from tube worm, I think, into E. coli, does this E. coli have any concentrating mechanism for carbon dioxide? Like usually native organisms have some mechanism by which they are going to concentrate CO2 and then perform the activity. So was the same done for your E. coli organisms?
A very good question. The question is on whether E. coli has a CO2 concentration mechanism. Actually not. So the thing actually we found is, I did not touch on that, but cyanobacteria, the Rubisco basically has two features. One is the catalytic turnovers, which cyanobacteria form is higher than the plant. But in terms of affinity, the Km, the affinity, actually the plant Rubisco has very high affinity, which means they can utilize the CO2 in the nature, which is present in very low concentration. So that's so in overall, the catalytic efficiency, which is the turnover number divided by Km, the plant Rubisco is not low, it's pretty good actually. So the cyanobacteria has actually relatively high Km, which means that they cannot utilize the very low concentration of the CO2 in the nature. Rather, they are using carboxysome, because the carboxysome in cyanobacteria can concentrate the CO2 into that. So that's the mechanism how cyanobacteria can grow in ambient temperature and CO2 concentrations. For regular E. coli, that's a problem. So that's why we have to feed CO2, high concentration of CO2 in the incubator to allow that the form 2 Rubisco. The form 2 Rubisco from bacteria also has a high turnover number and low affinity. This is also understandable because, as I said, the tube worm living in the deep sea, they are living around the surrounding the volcano region. The volcano region is rich in CO2. The CO2 concentration is high. So it's not difficult to understand why the form 2 Rubisco, isolated from the tube worm, will have a low affinity to the CO2. So when we transplant the tube form 2 Rubisco into E. coli, we encounter similar problems. That's why we need to have a high carbonate concentration in the medium, or we put the E. coli in a high CO2 environment to allow them to utilize that. So that's a good. But we also see my colleagues, some colleagues also reconstructed part of the carboxysome in the E. coli. There's a PNAS paper on that. So if carboxysome, which is composed of about 20 proteins, can be functionally reconstructed in E. coli, and the reconstructed carboxyl can entrap the Rubisco in that organelle, then that CO2 may be able to grow under the ambient CO2 concentration. Yeah. No, thank you for all the questions, all inspiring questions. Yes. Okay. Okay. Thank you very much. Enjoy talking to you. Bye bye.
you very much, Shyam, for this very nice and kind introduction. And I would also like to thank the organizers, basically the Reversition and Shyam, for inviting me, giving me this opportunity for this wonderful workshop of the host. So, Professor Inli gave a very elaborate introduction of the synthetic biology and what is the definition of the synthetic biology. And one part of it is he mentioned something like synthetic genetic devices. And, so one of the way historically the term synthetic biology came from the idea that if you can actually think about the biological system and the engineering system, and then you take the design from principles from the engineering system and try to develop some device in the biological. So that is one of the part of the synthetic biology, the way it actually started. And my talk has the flavors of those part of that synthetic biology. So when I was in my high school, there was a very an important news came, and this is Gary Kasparov, and probably you know her, all of you about him. So he's one of the greatest chess player of our time. And this is what this picture is actually showing, that Gary Kasparov has defeated by a computer, Deep Blue, made by the IBM. So this particular event and this particular year as one of the greatest thing in the computer technology, the computer science, because it's the first time you're seeing there's a fight between a computer and a human brain, and then and you can see what is the results. So this is also very important scientifically and technologically. So this is a tipping point, or it's a very critical picture when we start understanding that you can actually think about a computer which can do a kind of, definitely this Deep Blue is not a thinking machine, but you can play chess. But so to people, it looks like that the computer can think like a human and can do a lot of work which we believe that requires the brain, like the brain of the chess. So that is why it's a big start of the computer science in terms of the artificial intelligence. And but just to tell you that in 1997, Deep Blue is not exactly the way we understand artificial intelligence today. Okay, so that's a different topic we may discuss at the time. At the very same time, if it is a good thing for the computer technology, at the very same time, it is also for the biology. Because now people started thinking, because we know that we use our brain to do all kinds of computation, like the intelligence, we do not, and what we do, the imagination, our creativity, our feelings, and even the consciousness. Now, so at the first time, people started thinking, okay, so we can also try to understand the brain from aspect of computer engineering. So that is why it's a very critical juncture. At one point, you have some people are inspired to work on the computers so that you can look like a human brain. And now we start understanding human brain from the computer science or computer engineering point of view. However, today we do you know there are two different kinds of school of thoughts in terms of artificial intelligence. And one school of thoughts believe that if you can actually make the whole connectome, kind of a connectome like a human brain through the computers or some circuits, then you can actually think about what the consciousness is. So the computer could be the consciousness. So that is like 90% of this AI or hard AI people think about that. And there's another school of thought, which is minority, which is led by Professor Roger Penrose, who is a physicist, you know who Roger Penrose is. So he believes that a lot of human thinking, a lot of human things, what we do is basically not, it's non-computable. So for example, if you actually read his books like the Shadows of Mind or the Emperor's New Mind, so it's a very good explanation of that. I'm not claiming I understand that, but one point he mentioned that the understanding of the natural numbers, and this is one of the things which cannot be done computably. So please don't ask me, I don't know what does it actually mean, but I mean, I don't know what how the theories goes on that. But what he suggested that So this is one of the examples where you cannot do by computationally. So, but your brain is able to understand the natural numbers, so which is going to tell you that your consciousness is probably not computable. So that is a different areas of things. But as I said, 90% of the people believe that either you do consciousness or not, but definitely you can do a lot of the intelligence and a lot of things our human brain. Now, if you really want to develop a human brain, a connectome, the same as this, all connectome, the energy budget for this would look something like that. So 1 megaton. So 1 megaton is the energy which you can get from a mini hydropower generator. So, okay. So it's a huge amount of the energy. Now imagine this movie Terminator 2. So this is a robot. Okay. So if you want to make a robot coming from the future, either to save the human being or to kill this human being. So if you want to develop a robot, so kind of the energy, so like a mini hydropower energy we require. You cannot have on Schwarzenegger said a mini hydropower. So you can at this moment you cannot think about a robot exactly like who is looks like a human and work like a human, because that's of the energy budget. Now, what's the energy budget of the human brain? Energy budget is very small, just 20 watt. So sometimes if you see in animation, sometimes if someone gets an idea, you get a kind of the bulb. So I think that is a very interesting insight from the animator who first developed this idea. There's a thinking and it just give a click and there is always a bulb. And this kind of the bulb which we used to in our childhood, and this is the kind of 20 watt the bulb. So it's sometimes it's a very irony that who actually thought about this thing. So it's like a 20 watt. So this is the energy budget of the biological system. So in one case, so the point I want to make is if our brain, if you think of it as a computing machine, it is not only this enormous computing machine, but its energy budget is exactly so low. So it is very much energy efficient. So electronically or any other machine means, if you want to develop that, it is extremely difficult for this technological era. So now we saw that if you have this kind of system like the brain, that is a computer, it's a very interesting set of the computers, but that's interesting set of computers in the network level, because a lot of neurons, the billions of the neurons, the trillions of the neurons are making trillions of the connection to go there. Now the question is, if that kind of the computation, can you see bacteria or a very simple cell? And I don't know if anybody has have seen this movie. If you haven't, I'm not recommending it, but what I'm trying to say is that in this movie, this Amir Khan played a role where he forgets everything and he had to take a revenge. That is why he writes everything in his body, and that's the poster says. So in this movie, there is a scene or a sequence. In this sequence, Amir Khan was chasing a bad guy. So he was chasing, and suddenly one truck was coming from the other side and he hit the bad guy, and the bad guy probably died, I don't remember. But at this moment, exactly at this moment, Amir Khan forgot everything and he looked so perplexed in the middle of the road. So what this sequence or the scene of the movie tells us or teaches us, it teaches us that when you are taking an action, okay, so when you are taking an action, you have to remember just previous instances. For example, when you are chasing that bad guy, you are just chasing, and at the point where you forgot everything, you do not know what to do next. So to do any action, you must remember the previous instances. Now think about this bacteria, right? So we all in our high school studied a term, something called the chemotaxis. So if you have a chemical attractant, bacteria goes towards it. Now think about thought experiment. Now you have a beaker, and beaker has a full of water, and from one part of the water you give one some food, let's say some sugar crystals, and then the sugar crystal will start dissolving, and then it will make a concentration gradient. So at right hand side you have a higher concentration, and it is coming down with the lower concentration. And in this part of this beaker, you just put a bacteria. So it is thought experiment, think about it. Now, what this bacteria will do? Bacteria will swing towards the food because the concentration of sugar is increasing in this way. So it will start swimming. Now, when it starts swimming towards it, what will happen? There is always some turbulence in the water. So these water molecules actually hit the bacteria. Bacteria is 2 to 5 micrometer particles, and it will go in the other side. So when it's on the other side, if it starts swimming on the other side, then it is going away from the group. So what bacteria does at this moment, it actually stops and starts like spinning. So the stumbling motion happens. So bacteria has to take a decision, either it has to go swim or it has to stop and start spinning. So how they are taking the decision? And again some water molecules comes and give the turbulence, and again this bacteria goes in this way, and now bacteria again starting the way. So between taking this decision, first thing it has to sense the sugar concentration at the 2 time points, t and t-1, and then it has to decide at which way the concentration is higher or lower. If it is higher, it will take a decision to swim. If it is lower from the previous instance, it will actually stop and leave the contained motion. So bacteria has no brain, but to do that, you can exactly see it must have a memory. If it doesn't have this time level memory, transient memory, bacteria cannot survive. So how this memory is forming inside the bacteria? This memory is forming due to the interactions of the different molecules. So there's a lot of papers on that. So this is, people use control theory to understand this kind of chemotaxis, and they showed that how this memory is formed. It's very complicated. But one thing is very simple, that just through a molecular interaction, you can do memory kind of computation. So the computation, it is not only happening in the brain, computation is also happening in the bacteria all the time. So this is, if you think about the starting point, so one of the milestones of the synthetic biology, sometimes thinks that the 2000 is the starting point of the synthetic biology, because the 2 papers came, one from Gardner, who published in this Nature 2000. In the same issue, there is an oscillator also came from Ilwich, magnetic oscillator. So here, this is a very simple system. This is just a plasmid, and it expresses the green fluorescence protein. Now, what will happen if you give in this system? Now, we put the plasmid inside the E. coli, and if you give the IPTG into the system, you see the GFP expression increases. Now, you take this IPTG off from the system, and you still can see that your GFP is expression. Now, you give an ATC, another molecule to the system, the GFP expression comes down, you take the ATC out of the system, it still actually stays on. Now, if you see, this is very important. So, if you have studied any biology book, it's called microbiology, biochemistry, any simple biology, you will see this kind of thing. So, you give inducer, and you will see there is an increase in the concentration of a certain particular protein. But what is the difference between the book and these things? In the book, what you can see, if you take this inducer off from the system, the expression of that particular gene comes down. Whereas it is took off from this thing, but here it stays there. Okay? So it has a memory. So for example, here in my hand I have a laser pointer. So if I press here, you can see there is a light. Okay? If I take my hands from the switch, you see there is a no light. We call it switch, but this is not switch in real engineering sense. Why? Because it doesn't have a memory that I press at a certain time. Right? Now you think about any light switch of this room, or a light switch in your home, your hostel, or wherever. So if you actually press the switch, you see the light is turned on, you take your fingers off, still this light is on. So it has the memory that you given a stimulus, take the stimulus off, still it has the memory of the stimulus, so it is turned on. So that is the switch is in engineering the simplest memory device. So what they actually developed, these troubleshoot is the simplest memory device here. So now, how to actually develop that? So first, you need a qualitative principle. They took it from the RS latch, which is the electronic equivalent of a simple switch. And what is the biological manifestation of this RS latch? You need 2 mutually repressing promoter gene pairs. So when you know that, if you take 2 mutually repressing promoter gene pairs and you put it in the equalizer, you cannot make a switch. Why not? Because this will work as a switch. It has a quantitative parameter. So if you see the quantitative principle says only in a certain parameter range it will give the bistable behavior, which basically means that that point you choose your two mutually repressing promoter gene pairs. Now we have to engineer the system in terms of the transcription and translation of it, in terms of the protein DNA interactions, and then if you bring it to the bistable region, you are going to get this kind of the memory behavior. But if the parameter range out of this question, you will get the more stable behavior like this. If you give this IPT, you take the IPT off from the system, it is coming down. So, okay, so that is why, so this you can understand that this has a little difference between the conventional genetic engineering and this. So this is basically a design-based principle what we do. Okay. Thank you. Yeah. So you need this kind of the design principle to develop something like a memory device, a very simple memory device. Now, this is when you think about this is the starting point of the synthetic biology, but the whole idea of the synthetic biology is probably coming from the systems biology. So in the last 3 days, probably you have studied the synthetic biology a lot. You know what is the systems biology. So I don't want to touch that, but just from my perspective, a synthetic biology perspective, the way we think of this, perceive the systems biology in this way, that so any biological function, it does not depend on a single gene or protein. It depends on the interactions of the genes and proteins. So that's why if you take a cancer cell and a healthy cell and you do the transcriptomics, and then you find that thousands of the genes have been upregulated, thousands of the genes have been downregulated, and you are completely perplexed and which pathways, which genes, that is why you don't get a marker for a particular cancer. Okay, and so that is the starting point. But interestingly, what I say that it's the interaction of the genes or the proteins giving this biological function. This interaction is not very simple interactions. Mathematically, they are all nonlinear interactions. They have a lot of feedbacks and there is a lot of noise. And it is so difficult to understand mathematically or even physically because we don't have the technology or the mathematics to understand all those things. So what we can do, so we look at the human knowledge base where the people work on the networks, and that is basically the electrical engineering or the electronics engineering, the computer engineers. They always deals with this kind of interactions. Though those interactions are definitely much less complex than the biological interaction, but still they deal with the nonlinear interactions, feedbacks, and the noise. So one idea or one of the philosophy of the systems biology, if you actually take those principles and try to use in the biology, provide understand the biology at the systems level. So if this philosophy is correct, the other side of the coin of this philosophy is why not then we actually develop some of the synthetic genetic circuit, taking these principles from the electrical engineering. Now, these small circuits we put inside the cell, so you see that the change of the function in the cell will develop a new function. So if you see the hierarchy of the engineering in terms of the computer engineering, so you start some physical layers, then we connect the physical layers to make some logic gates, and those logic gates are connected to microcontrollers, and these modules will go to the computer and so on. And you have a very well established design principles how to do that. Now, we come to the biological hierarchy, you start with the protein and genes, and you develop some of the biochemical reactions. Okay? And then biochemical reactions goes pathway cells. But however, this evolutionary pathway, this evolution which is tuned for the evolution by 4 billion of a year, we do not understand very clearly how this protein gets connected to the biochemical reactions, to the pathways and so on. And definitely we will try to do that, but it will take much more time to understand the real biological principle of they are connected across the scale. For the moment, what we can do, we can take the protein and genes and develop some reactions to work as kind of a gates. In the logic gates, in electronics, your input and output is the flow of electricity. But when you come here, your flow of your inputs and output states are different kind of the chemicals, metabolites, proteins, and so on. So we develop a biochemical reaction which works like a gate. But now we know the principle how to make a module from our different function from the gates. You connect those biochemical reactions in such a using this principle, So you get a pathway which will give you kind of a device function, and then you connect and so on. Probably you get a very completely new kind of the cell, new kind of the function. And that is the motto of the synthetic biology for the last 20 years or so. Now, what people actually develop? People develop a lot of different, all kind of logic gates, the NOT gates, the OR gates, NAND gate, and whatever they are, and different molecular mechanisms that they put, and they develop a lot of different kind of logic gates. Now, why this logic gates are important, and why this engineering principle is important to actually design a new biological function. So this is important because, for example, so all of our computers have something called the adder or subtractor, and subtractor which actually adds number and subtractor. So a lot of adders, full adders are connected together to make the whole adder unit or subtractor unit. Now this is a full adder truth table. So, and how you want to make this truth table? You have a different logic gates, you connect in this way, and it will give you the adder. Now, if I tell you that you have to make a system where your ABC is some inputs, some chemical inputs, and your other chemical or biochemical outputs are the C out and the rest, and I want to make, can you develop this kind of things by thinking? By thinking, even for the best genetic engineers of the world, it is almost impossible, because you cannot think that how these three chemicals will go through a network and then it will give this behavior, how this connect to that, how this connect to that. You cannot, just by thinking, you cannot do that. So, what about if you develop some biological gates, right, that you can do by thinking, and then you connect following these principles, then what will come out of it is basically an adder. So basically, it will follow this kind of, and you do not have to understand that how in between how this molecules is going to work with this, or how this feedback. You don't have to understand, and that is the motto of an engineering, to make things simple, streamlined, and so on. So through that, what people are able to do just in 2018, a full adder in using mammalian cells. They have a 9 different kind of the mammalian cells engineering. Following this kind of principles, you can see here, and they are connected with different kind of the hormones and the chemicals. One cell connect with another, and they are able to make this kind of this is the adder. So that is why it is important if you want to develop a function. This kind of engineering principles of logic is important. So there's some other examples I can give. So here they developed something called a classifier circuit. So this is a microRNA based synthetic genetic circuits. Now, if you have a cancer cell like the HeLa cell have a special kind of microRNA signature, not from a single microRNA. You can see 1, 2, 3, 4, so 6 different microRNA, and 3 of them actually upregulated, 3 of them are downregulated in the HeLa cell. So now you develop a classifier circuit which actually can recognize this signature. So they put it in the HeLa cell. When it matches with this signature, the circuit actually starts producing a protein, which protein actually forces this cell to go to the above process. Wherever you put this circuit to the normal cell, the microRNA signature won't match with the things. So what we can identify, and there is no match, there is no effect. It is just says that. So this is another kind of interesting example of what you can do. And another interesting example I would also like to, so that's a program multicellular communication. So what are those? This is nothing but the bacteria are plated on a petri dish. Okay, so bacterial lawn which can produce 2 different kind of the proteins, either red fluorescence protein or the green fluorescence protein. Now it is not like that I took a green fluorescence producing bacteria and just stick in this way, and then I take the red and take it this way. No. This is the whole lawn of a single kind of engineered bacteria, which has the potential to produce both the green and red fluorescence proteins. And now, if you give the inducer, based on the inducer concentration and the communication between the bacteria, this kind of the pattern formation has happened, and you can actually simulate this pattern formation from beforehand. Now, within the 5 of them, one of them is a computer simulation, other 4 are the experiment. But just looking at it, you cannot tell me that which one is the computer simulation and which is basically the actual experiment. So this is happened in 2005 from around my group, and you can see whatever we can only make 2 concentric cycles. Now if we go a few years down the lane, another group from China, they actually developed many concentric rings like that. So what is the importance of this? If you take this is a Drosophila embryo, and when in a developmental biology, all of us started our life from a single cell, and then it goes to the 2 to 4 and then 8, and then this started actually making different kind of asymmetry. So even in the Drosophila, when the segmentation happened, you find there is this kind of the patterns formed by different genes like that. So what we are able to get a similar kind of thing, even in the 2-dimensional, this is in Drosophila is a 3-dimensional, 2-dimensional something like that. So we started understanding how to develop this kind of the pattern formation. So one day people have also started in the mammalian system. So one day probably we actually develop the organs by engineering, hacking those kind of engineering principles what already have in our development systems. So with these 3 examples, there's a lot of other examples we can talk about. So I just come to in my lab's goal. So one of our goal is, can we make bacteria artificially intelligent? So the artificial intelligence is basically now there's very few words. The 90% of this artificial intelligence or more are basically the basic idea of this artificial neural networks. So what is artificial neural network? The idea of the artificial neural network is definitely coming from our brain. Our brain's neurons are connected and give this function. We know a lot of molecular details of the neurons, but we do not know how this neuron, when these neurons are connected, how it gives us the imagination, how it gives our feelings, and so on and so on. But what we know experimentally, that if you take a neuron and give some inputs here in terms of x1, x2, xn, and then you get an output in y1, y2 and something, then there is a mathematical relationship which is always matrix. And now you want to take this mathematics, so just you draw in a which is named then as artificial neuron. You give some input x1, x2, x3 and something. Now how this will behave? The input will behave in such a way that first this input will linearly combine, and when it's linearly combined, it is giving this your inputs which multiplied by something called weight. So x1, so the weight will come x1w1 plus x2w2 plus x3w2 and so on. So this, after summing this, whatever the function is coming from this summer, is now going through a function. There's a linear function also, but mostly it is a non-linear function, and it gives the output. So this is if you take any neuron and it will behave like this. Now you connect this kind of whatever your paper neurons with other neurons, like other developed same mathematics, what will come here is called artificial neural network. It looks very simple, but it can do a marvelous job. For example, think about the Facebook. Let us take a photograph of this our whole symposium and put it in the Facebook. If you do that, the Facebook will basically going to tell you, do you want to tag this guy, do you want to tag me, do you want to tag you with this and this. So how Facebook is identifying our face, they are using this kind of artificial neural network in their server. So a face recognition server have like 70 to 80,000 different kinds of nodes. It is not that simple thing. But what is happening? So if you give photographs, it takes pixels from your face, and these pixels are basically these x1, x2 inputs. And when you have these inputs, what you're going to do is, so first, so they have a lot of W's, because mathematically, there's just mathematical softwares, all these W's they calculated, and whatever the output they will give, this output won't match with your face or anything. So what you do, then is start changing these W values at various points. Okay, it is not random because they have a lot of different kind of algorithm how it will change this W. And you get an output. This will match with the previously given uploaded picture of yours in the Facebook. So that's how it is happening. So physically understanding what is happening there, it is very difficult, but mathematically it is happening like that. So now again, if you think about all this ANN people do by software, by the computer, and probably 95% of it or more, right? Now again, if you want to make a robot, okay, and if your server is in the US and the Facebook server, and here is a robot is going to do some work, it's very difficult, right? So what you require that you just don't get ANN software, but you need ANN hardware to actually make this robot. So that's why people are developing a lot of chips. It is called the neuromorphic chips. The idea that in these chips, what if we call it a circuit, this circuit is not following this hierarchical electronic principle, the way gates are connected to that. Instead, they are following an ANN. So people are developing a lot of new chips for that. People show there's photonics is the possible. Even people show that in vitro DNA computation actually can show some of the ANN. So my lab is asking this question: can you make a bacteria as an artificial neuron, and then connect a bacterial culture where the bacteria connect with the other bacteria and actually develop an artificial neural network to solve some of the computational problem? And we are very happy to say that very recently we were able to develop the first artificial neural networks with any kind of molecular niche, either bacteria or cell or any other cell. Definitely our neuron is a neural network, and that is definitely. But this is the first artificial neural networks with engineered cells we developed. So to do that, how we chose, the very first thing what we chose is we chose a decoder and encoder. I mean, this is just giving a two example, because decoder and encoder are very important in any kind of the communication devices. Either it's a walkie-talkie or it's a system like aviation system. So I just wanted to, decoder and encoder is a very important system, and we have to develop decoder and encoder using ANN, using the bacteria. So if you take the 2 to 4 decoder, is a very simple decoder, which its electronics diagram looks like that, and its truth table is something like that. So if you have 2 input X and Y, so you get 4 different combinations, and for 4 different combinations, you can actually decode that signal in 4 different ways. So this is you can see this, the 4 different decisions can be made. So we do not want to develop this electronic structure through these biological gates, but we want to develop ANN. So here, first we map this whole system into mathematically like this. It requires 4 different types of artificial neuron, and now we engineer this mathematics. It is simply we engineer the mathematics inside the bacteria by changing different molecular interactions. So now we have 4 different kinds of engineered bacteria, right? These 4 kind of engineered bacteria mix in a conical flask, and you give your problem through the chemicals, and what you see is the answer here. So IPDJ and ATC, based on these combinations, you can see it produces 4 different kinds of fluorescence proteins. And if you see here, this is fluorescence only on the diagonally, and everywhere it is no fluorescence. If you see come here, that it gets 1, 1, 1 diagonally and 0, 0, 0 everywhere. So what I try to tell you that so we are able to make this kind of the system using the artificial neural network. Now I'll tell you that how we are going to do. Very first thing what we develop is set of robust rules that anybody can follow. So that if you have a computational function in terms of the truth table, we develop a rule that how this truth table is mapped to a neuron. So this truth table probably can bring it back to the 3 neurons so that you connect them and you get whatever the answers are here. So this is the set of rules we develop. So when you actually coming down to a single neuron like this, then we also develop the molecular principle. So take this mathematics, what would be the molecular principle from this mathematics is going to tell you what kind of circuit or what kind of genetic system you have to develop. So that's when you develop these 2 things, we started building the circuit. So first thing I said, we actually develop 11 different neurons or vector neurons, what we call vector neurons. Now, from this library of this 11 vector neurons, just mix and match, we can generate 7 functions that we have actually shown here. I just shown the 5 of them. So basically, it's a demultiplexer, multiplexer, majority function, a decoder, and encoder. So this is the first from this how the truth table. We do not need this electronic principle. We just from here we can map to this ANN. So when we actually develop these rules, we start, as I said, started building it. So for example, here is one of the vector neuron, name is vector neuron 1. To develop that, we develop a lot of different kind of the synthetic promoters. Because see, the rule says you have a mathematics and you have to make a molecular engineering of that, so that that molecular engineering follows the mathematics in terms of the inputs and outputs. And to do that, you develop genes and proteins. There are a lot of transcription factors connecting with the promoters and do all these kind of things. So in the synthetic promoters, you take the Tata box sequence, and then in between you start putting in different transcription factors in terms of numbers, in terms of where you want to put. And it is exactly happen like the Facebook. In the first try, it will never give you the correct behavior. So then you have to change the W values. So W values means which is we do by different kind of molecular engineering by controlling the transcription and translation rates and changing the DNA protein interaction, changing the copy numbers and so on, so on, so on. So this is a very busy slide, I'm not going to explain it, but when we actually come to the final things that where all the molecular and genetic reactions which have the appropriate parameter range become something like that. Here you can see this is a simulation for this particular vector neuron and this is the experiment. So this is a good match. Similarly for this vector neuron, this is simulation, experiment. Here this is simulation and experiment, and they match very well. So this is our final vector neuron. So now your vector neuron library is ready. So we have, as I said, the 11 different vector neurons. You now, according to the design, you just plug and play, make a culture, and give the problem, it will solve. So here is, you can see this basically the encoder, which is a little more complex than the decoder, and this is the actual result. This is the things. Okay. So we just showed that okay, we can actually develop that. We have developed something else, but we also try something else, and this is called the reversible computing. So reversible computing is proposed by Richard Feynman along with Fred Dean and Tropoli, so many people together. Today, when another technology people are trying to develop is that quantum computing. We all heard about it. I do not know anything about quantum computing. Only thing I know, in quantum computing, you cannot use the AND gate, NOT gate, whatever the irreversible gate we are using. You have to use something called a reversible computing, like the Feynman gate. So reversible computing has two particular importance. One characteristics. One is the thermodynamic reversibility, and one is the logical reversibility. What that means, logical reversibility? So if you have some input, your output you will get back. So it's from this output you can map the input. So it's a one-to-one mapping. So 0, 0, 0, 0, 0, 1 to 1, but 1, 0, okay, it's diagonal, but it's 1, 0, 1, 1. So this is a kind of thing where from the output you can map the input, which basically means no information is getting lost. That is logical reversibility. And second, it must have some thermodynamic reversibility. What we know, a biological system, what we are working on, any living biological system, is definitely an irreversible thermodynamic system, right? We cannot attain reversibility there. However, what Richard Feynman envisioned that if you do the reversible computing in the biological reactions, the biological reaction takes a very low amount of energy. I've seen the brain, right, 20 watt compared to the 1 megawatt, right? So biological reactions require 6 orders of the instant, it was 6 times less energy than the transistor, what is that time transistor nowadays. So you cannot attain the thermodynamic reversibility, but its energy expense is so low, you are actually going towards this kind of the thermodynamic reversibility. So and people develop using biological reactions in vitro, Feynman gate and Fred Dean gate. What we try to develop is inside the cell. Okay, so we using this ANN, we are able to develop the Feynman gate and Fred Dean gate. Both are the two important gates in the reversible computing. And this is the principle, and then this is the result. So we are happy with that. Now, so what is the importance of this particular work? Because for last 20 years, when we are thinking to develop synthetic genetic devices, we have to use this kind of biology. We introduce a new computing system, and that is the ANN. So why I believe that ANN is much more important? Because there is ANN, the mathematics of ANN is coming from the universal approximation theory, which basically says that if you have a simple neural network with one hidden layer, it can approximate any continuous function within a specific parameter range. Means you can actually develop any functions through this ANN, and our goal is to use this ANN into synthetic biology to use actually different kinds of function. Now, this is the starting point, and I'd like to show a very recent, because the result came to me yesterday. My student actually sent that, and this is so. If you do the ANN, can you do something which you cannot do using those kind of electronic principle? So I believe yes, that's true. So in 2015, Hidama's group actually developed something called a half adder in the vector gate. So you can say this is the half adder. So if you connect two half adder, then what you can actually make a full adder. So it's a single bacterial cell, then in this half adder, and you can see this how many genes you have to put, and all these things have to work in a temperature parameters in parameter space. And so this is the first half adder. And in 2018, the ETH Zuri could make a mammalian cell full adder. Now in mammalian cell full adder, as I say, it is not possible in making in a single cell. So they have 9 different cell systems. So from one cell they make a part of this circuit, another cell, this part of circuit, this will probably give some chemicals here, this is some chemicals, these chemicals will take by the other cells and this go on. So this is a kind of distributed computing and various kind cell. Now problem is, you can understand that in a single cell making a full adder is not possible, okay, because we don't have that kind of capability at this moment. And if you want to do this kind of distributed computing, people actually showed that it's possible in mammalian cell, but this is not possible in the bacteria. The reason, because you need 9 or 10 different kind of chemicals which are connecting to 9 different kind of cells, so 9 different orthogonal proton sensing molecules which will connect, and this is extremely difficult. Even getting 9 in bacterial system is extremely difficult to actually that. So that in bacteria nobody could show a full adder system. So here what we believe that we can develop not only the adder but also the subtractor, which means that this bacteria can add and subtract binary numbers. If you use this ANN practice, we do not need 9 layers. We just need 1, 2, 3, 4, 5, 6, 7 kind of the cells in a single layer, and this is not going to give another chemicals to other cells. But if you actually put in the single layer, we can able to make this bacterial full adder and this bacterial full subtractor. And yesterday we got this our preliminary results to this full adder. So if you don't understand, I just want to tell you the adder looks subtractor look good, adder is not looking good because there is some leakage. Specifically these two should, there should be 1 and this should be 0, but it doesn't look. This one should be 1 and this looks black, but it is not happening. So we believe that we will actually do some engineering on that and again reduce this leakage from here. So we'll see that. But this is a preliminary result we just got yesterday, and I think we are able to try to convince you, or trying to convince you that if you use this ANN in the synthetic biology, you can do much more things, much more which cannot be done in the electronic circuits because due to some sort of practical difficulties. So now a second problem which we will work on. So here is a maze problem. So any child, because it is taken from a child's magazine, so any child can actually solve this thing, okay? And if it is a special kind of child like the Harry Potter, they have a different kind of maze. Tweezer maze, if you have seen Harry Potter and Goblet of Fire, you probably have seen how to solve it. Now interestingly, if this kind of the simple maze, either is a muggle and normal people like us, and if it's a wizard like him, solving this kind of the maze are easy for them. But if you give a computer or a robot to solve this kind of a maze, this is very, very difficult because the computer has to solve this through some computational algorithm. We do not know how our brain actually see the lines from here to here. That is important. So first maze solving computer actually made here in 1971, and look, this is so much cumbersome machine. Now what we think that okay, so why not we make a biocomputer with the bacteria E. coli which can solve the maze. And what we have done, we've taken the simplest maze possible, and that is 2 into 2. So in 2 into 2 mazes, you can have 16 different problems, right? Now first, when you have a maze, so how do you computationally represent this? So any maze you take, it has a problem matrix and a solution matrix. The problem matrix, if 0 means there is obstruction, 1 means it is open. So here 1 means it is open structure, and anything is 0. So corresponding to this 16, they have a solution matrix. In solution matrix, if there is a solution, the path, you will get 0, 0, 0. If you knock the path, then you get some 1. So what you have, if you take this problem matrix and the solution matrix, we map this in terms of a truth table. So in this truth table, this problem matrix will be present with 4 different chemicals and solution matrix with 4 different process proteins. Which basically means a problem matrix. If you have a chemical, like say, and if you give these chemicals to the media, means it is 1, means there is a path, and if you don't give that chemicals, it is 0, then there is no path. Okay, so we map it in the truth table. So this is very cumbersome. I mean, in terms of the synthetic biology, it is very a difficult system. So we able to use the concept of the distributed computing, and we design 6 different kind of the cells. We actually work as this particular computer, and we engineered the 6 different kind of the cells. I'm not going to the details, but anyway, when we mix these 6 cells together, that is your biocomputer or the main solving computer. Now here, if we study all this computation, and in 13 of the cases there is no solution. Okay, and 3 cases there are solutions that we can see by the Proton thing. So we are very happy. We are much more excited because this work has been featured in the MIT Technological Review, and many people actually commented on that from the fields, and so we are very happy. At the same time, it is featured in many other international media items. So now we want to see, go beyond bacteria. Why you cannot develop some of the computational system where the bacteria and mammalian cells, the cancer cells, they will interact each other. So what we actually first developed E. coli, which cannot attack a cancer cell, but we engineer that it can attack the cancer cell. This is nothing new because people are actually developing this for a long time. So here we produce that circuit, and again later we can talk about the circuit how to make. But this is something not we have to develop. Many people actually develop that, but it is in our lab. We have seen that when you engineer this E. coli, so you see this is the HeLa cell and this is in the pin-like structure. The pin-like structure is the E. coli which is attacking the cancer cells. Okay, and now if you just co-culture these HeLa cells with the non-engineered E. coli and you wash it out, you cannot see any bacteria is attacking something like that. So when our invasive bacteria is ready, we made a Feynman gate. In this Feynman gate is very recently published in essay Synthetic Biology and featured in Nature India. So in here we give the input to the bacterial cell. These inputs are getting computed by the bacteria, and that computed information is transferred to this cancer cell, and then another computation is happened here, and by this computation the two native genes of this HeLa cell got silenced. So but this silencing is happening based on the Feynman gate principle. So it's an intercellular Feynman gate. So what you can see in this result is that you have input chemicals you apply to the bacteria, and you get the output which is manifested in the gene silencing in the HeLa cell. So it's an intercellular Feynman gate. And so the question is, if you ask that why do you really need this kind of the computation using the bacteria or the mammalian cell? At this moment, there is no computer or a robot available in the micron range. Right? There is a term called microcomputer, there are terms called microrobots, but they are at a centimeter, the 3 centimeters, 5 centimeters kind of that. The reason you cannot bring it to the micron level because it requires energy. You cannot bring a battery in the 1 micrometer or 2 micrometer level. So, which basically means there is an untapped potential in this the micron region. Now think about a bacteria. A bacteria is exactly 2 to 5 micrometer. It doesn't need a battery. It can take the food from the outside environment. Now, if you can develop computational function with this kind of the living cell, then what can happen? You can actually develop a computer in the micron level, and it can have a lot of, as I said, some type of potential in the medicine. Probably is also in the bioenergy. I do not know, but think about a reactor where a lot of consortium is working, but those consortium is highly engineered, like they interact with each other and as a computation. So bringing the computer exactly the producing probably the bioenergy on the reactor or something. So I don't know, but those are the possibilities. So that is why we are excited to in this field, and we want to actually increase this field with how complex kind of computation we want to develop. Might have another 5 to 10 minutes. Yeah. So this is not part of the title of our, but this I sometimes feel you might get interesting because this is another area where synthetic biology is trying to phrase, and this is a space synthetic biology. So people do know that. So for example, if you want to go to the Mars, okay, and want to make a research base, it takes like a almost 1 year to go and 1 year to come back, 6 months to 1 year, and then you stay here. So you cannot take that much of the resources there. Okay, if you want to have a biryani, you cannot take it from Delhi, right? We have to cook it there. Not only the cook it, you cannot take the rice and the mutton and the Bengali biryani, it is always aloo. So you cannot take all these things from here to there. You have to grow everything on the Mars, and it is called the in situ resource utilization. Starting from your medicine, your road, your habitat, your medicine and materials and everything, you have to make on this thing. So that is the plan. But the conventional biotechnology cannot do that because it is not that robust. But that is why people do believe that if you can develop synthetic genetic organism, and now you need only few vials, you take it through your rocket and put it there, so they will do all the tasks for you. So this is the one idea of this. They believe that the synthetic biology will be the key to the space thing. So what's our goal was that can you develop a microgravity biosensor? Okay, so that when you go to the space, this microgravity biosensor can work in your way. So for this we took a long time, because very first thing when we tried to find out that if there is any gene in the different kind of the bacteria which will affected by the microgravity, and then found there is a fundamental problem there, because microgravity is very low perturbation, so you always get like a 20%, 10% increase. And you know, if you do a transcriptomic study, if there is a 10% or 20% increase, you just have to put them out. Only something which is more than 50%, more than 100% increase, like 1.5 times or 2 times, those kind of the genes you are going to take. In cancer cell, that's not the problem. It is 10 times, 1,000 times, those kind of outcomes. But in microgravity, it's very low perturbation. So you actually lost the basically very different kind of the pathway. So I am not showing here, that is not the part of my talk, but the very first thing what we did, we developed a systems biology pipeline through which, using a lot of machine learning algorithms and the challenges was this analysis of the genome-wide data of human cell space did not tell much, and our research actually overcome this limitation. From this, the things we able to identify a lot of different pathways and able to explain, that is computationally a lot of unexplained facts. But from there, we also identify, because we take all the space data available, transcriptomics data available in the world that time, okay, through the NASA or European space agencies. What we able to identify a couple of candidates which we able to use for our microgravity sensor thing. So what we found, there is something called HFQ protein which is get deregulated in the microgravity. How, we do not know, but that we are confident enough that it is happening to different bacteria in different data and different pathways. So that is why I took this effect, and this HFQ is instrumental to actually when you want to use the siRNA connection with the siRNA to stop the any expression. So we developed the synthetic siRNA circuits which will take over this HFQ things and develop a circuit. And now here we have a microgravity simulator in our lab. We can actually, this is cell culture reactor. You can actually develop the gravity here, microgravity here, and this is in these conditions it will give you normal gravity. And what you can see here, the fold change between the degradation of the enhanced green fluorescence protein between the microgravity and the non-gravity is high enough. So this is the first microgravity biological sensor we developed, and this is the control experiment you can see. But it is not just the sensor. We can take that sensor and we connect potential to any biochemical pathways. So we actually chose a biochemical pathway of the cell division. FTSG is a kind of a protein which helps to cell division. If this FTSG is get deregulated, then what you will get this kind of elongated shape. So we engineered this cell, which is a defective cell, not gravity. Now we connect this, we connect our synthetic genetic circuits through siRNA to this FTSG protein. So now this cells, if we put in the microgravity, see, it is got cured. So we just showed that okay, you can use the microgravity as a physical signal to do some biochemical works. And with that, I like to stop here. This is my group. We like to eat foods more than we do work, and this is our finding. Thank you very much for your attention.
is not absolutely clear because I'm from a typical biology background. So I had a few questions. In one of the slides in the early part where you had designed the 2 to 4 decoder. So can you please go
Yeah,
back?
sure.
Yeah. So here, I believe you have designed 4 different bacteria where in each of these bacteria you have designed a particular neural network, right? So what I observe here is that a few outliers are there in each category. So my question is, so why is it not absolute? And if I take one outlier bacteria from each category and then subculture, what is going to happen? Will they continue to give the outlier expression or they will come back to the normal expression? So for instance, in the last panel, instead of M venus, some of them are expressing MTFP1. So if I take one bacteria from MTFP1 and continue to subculture, will they give M venus or they will give MTFP1?
Yeah, this is a great question, and the first the answer to your first question is it is human design. It is done by my students and myself or any of you or anyone. It is not designed by the nature. So in biology, there is nothing in reality. There is a 0 and 1. Not even in electronics, right? So if you this is the electronic decoder, if you have a very good instrument to calculate the electrons. What you see is the 0, 0. It is not 0. You will get some kind of the things, right? And here, from a synthetic biology point of view, though outliers is there because there is a leakage, and leakage problem you are getting, or anybody who is working, this is a bit. So this is a leakage problem. So the question is how much you suppress the leakage. That is a very fundamental question. So here I'm making those things. It takes it to. If you go this slide, oops, here I think this slide is about more than 3 years of work. Okay, so because optimization is a very, very difficult thing to do, and what we believe, these outliers for the practical purpose of this decoder is okay because this outliers is not affecting the things. But there may be some application where even this outliers is going to affect the things. Now, when you are coming to your second question, that if you actually take a picture neuron and you will see what you are going to basically see. So basically, when we have to design and optimize, we always design optimize as a single vector neuron, and we always see there is a leakage. And many cases there is leakage, many cases there probably is not. Now, so if you take the one thing, then whatever the outliers you see, you will be able to see that. But what we are excited that when you mix them, then nothing else is happening. It is not much more overlap. What we got in the single, we are getting it. So that is our part. But yeah, so someday somebody actually reduce this whole expression of leakage. Thank you for your question. Yes, please, right.
Interaction between mammalian cells and E. coli.
Yeah.
So you had shown 2 figures of HeLa, one with engineered E. coli, one without engineered E. coli. So, how much can you confidently say that it is not compromising on the physiology of HeLa cells? Because on the left hand side, I already see that the HeLa cells are not showing the appropriate morphology what it has to show. Like this one is typical HeLa morphology with the neural network-like appearance, but here I do not show.
Yeah.
So don't you think that by doing such engineering, it is compromising on the original property of the?
Yeah, absolutely. So it do that. So one way to see that how robust this system is. So you do this experiment and then you watch the EJP and the E. coli and everything, and then you grow the cells again and you see that how many hours it goes. So we went till after the infection we go for more than 4 days, and the confluences are okay. I won't say it's exactly. So definitely it is going to change the physiology and everything. So one of the, so we make from a intercellular computational point of view, but one of the reasons people wanted to develop this kind of the bacteria for the cancer cell to kill the cancer cells. Okay, and not only kill, because so what I tried, so you can actually silence a very particular gene, and when we actually do that, we also measure the genes which are related to that particular gene, and we haven't seen any change. So in that cases, I will see it's okay, but in terms of the physiology, what see, yes, definitely there is a change, but it will go at least till 4 days with proper confidence.
Hello, sir.
Yeah,
Thank you, sir, for such a wonderful presentation.
thank you.
Sir, I'm not from this area, what you are representing here, but it was really very, very interesting to know. It is the first time even I saw this area exists. So, my question, if I may ask you, how did you land at this interesting area? How did you, and how do scientists like you envision such questions and problems which generally we don't even think in that? We don't even know how did you land at this problem?
I can tell my experience. So I was a chemical engineer, and I got interested in the biological engineering by reading a book, and that is the Michael Crichton's Jurassic Park, and that's really why I really changed my whole career from a chemical engineer. Instead of joining a job, I joined the research, and the research on the biological engineering, not on the chemical engineering. Later I found there is a lot of people got interested on the synthetic on the biological engineering just from the Jurassic Park, which includes my supervisor who was a space engineer who developed that algorithm for the US robot transmission. He developed the algorithm for robotic communication, and when he was actually listening to a talk by Jim Collins, whose paper I showed, that published his paper, and he found that when Jim Collins was talking about some genetic interaction, my supervisor Dave found, oh, it's exactly the same algorithm I developed for the robotic communication. So there is a very natural, so he jumped into this thing. So, and I found a lot of my colleagues, they got interested in the biological engineering from Jurassic Park. So one of the guy who a Taiwanese guy who actually sequenced the genome of the mammoth, right? And he literally, we have got interested, but he literally took, I have to make a dinosaur. But he couldn't get the dinosaur DNA, so he looked for the mammoth DNA and sequenced these things. So there's a lot of people actually from there. Now, when you are when you are mixing the biology and the engineering, you understand there is a huge amount of the potential. So possibly we are coming from a completely different background. Okay, myself, chemical engineering, I do not know anything about electronics engineering other than I just read some to pass my exam things, and then I do not know anything about biology. So I believe that gives us a very fresh perspective from a very different aspect, and probably from the day it gives a different perspective came from those things. I think when these perspectives are coming into there, then there is a problem. But one thing, as both of you are trying to tell, I am not from this background, then I am also not from that background. Or okay, the whole question is when I joined this, I thought that doing this pipetting and this molecular biology, everybody say, oh, we are going to do molecular biology, it's a black magic, and many times it do not work. And I was then I thought, okay, I'll do my PhD in computation. And in a conference, I found there was a student who is a computer engineer and doing those kind of, who was actually showing this molecular biology. Then I think, okay, computer engineer can do, the chemical engineer can do pipetting. So I think the one thing is the barrier. This barrier, we should not believe there is a barrier, because so my students are all biology students. The mathematics they have done. Even I do not want to name him, and probably he didn't know how to add 2 plus 2, but he did some kind of these calculations. Okay, so I think the whole is that whenever you think that there is no barrier, whole platform is yours. I believe, and then you can ask different kind of the questions and different kind of things. That's my perspective.
Thank you, sir.
I just probably when Sam, there's a two different kind of stocks. One is intelligent bacteria and there's the. I think one good thing of the synthetic biology, because I cannot take both space and swim. This same synthetic biology, same molecules, same things, but you can have a very different kind of the application with the same expertise. I think that is one of the interesting part of the synthetic biology, and it looks completely different, but it's not at all. That is a sensor. So if you want to make a computation system, you have to develop the sensor. The IPTJ and ADC sensor is the same as the microgravity sensor. It looks completely two different fields, but that is why the synthetic biology is so exciting. It can actually affect many fields.